Selamat pagi, good morning Bapak Ibu sekalian, and good evening to Professor Zinky Weng from Boston. Are you already in? Okay.
Yes, I'm here.
Okay, it's great to see you, Professor Zinky Weng. And then I would like to ask check Dr. Sri Fatmawati from ITS Surabaya. Are you already in?
Yeah, hi.
Hi.
Good morning from Surabaya. Hi,
Yeah.
hi Jinky.
Yeah. Okay, I think we wait for another 2-3 minutes to allow the joining of the participants. I think then we will start directly of the event. Mr. Miftah, host, could you please share our agenda for this morning Indonesian time? So while waiting the other participant, I would like to start the meeting by saying Assalamualaikum warahmatullahi. Very good morning, Indonesian or Jakarta time, and good evening, or Boston time. Yeah. So we are having 12-hour differences time. Yeah? So I think maybe convenience for both of us. Yeah. To be together to see the today IPB talk. Yeah, ladies and gentlemen, allow me to moderate and as well as to open the session of our today IPB talk. So this event is realized due to the so-called MIRA program. MIRA stands for MIT Indonesia Research Alliance Program, which is managed by Institut Teknologi Bandung and funded by Ministry of Research and Technology, Agency for Innovation, or BRIN, Ristek BRIN. So the collaborators for these programs is Professor Zinky Weng from Biology, Whitehead Institute, MIT Boston. Myself, Iskandar Siregar from Faculty of Forestry and Environment, IPB University. Professor Heri Hermansyah and Professor Misri Gojan from Universitas Indonesia, and Dr. Sri Fatmawati from ITS Surabaya. Without further ado, yeah, I would like to invite Professor Zinky Weng to present. But before that, I would like to show a brief CV of him. Miftah, please. So we make a brief CV based on your website profile. So let me introduce Professor Zinky Weng. He is Associate Professor of Biology and member of Whitehead Institute for Biomedical Research. We are the three, me, myself, and Dr. Sri Fatmawati and Professor Heri Hermansyah visited the Professor Weng lab at MIT Boston last year. So before the pandemic, yeah. So early December 2019. So we are happy to meet at the time, and we discuss about our joint proposal. And luckily, the startup proposal was funded, yeah, by the committee. Yeah. So his research field is chemical biology, studying metabolic evolution in plants, as well as plant diversity for new commodity chemicals and medicine. His education is PhD from Purdue University and bachelor's degree from Zhejiang University, Hangzhou, China. So he received numerous awards, such as Beckman Young Investigator Award 2016, Alfred Sloan Research Fellow 2016, Shirley Scholar 2015, Pew Scholar in the Biomedical Science 2014, as well as American Society of Plant Biology's Early Career Award 2014. That's a short introduction of CV from Professor Wang. You may contact him directly after the meeting, and we encourage that our collaboration is broadening because Indonesia is so huge country. Western part belongs to Sundaland, and the eastern parts is belong to the Wallacea region. Yeah, so biodiversity is the key issue in Indonesian development agenda. Yeah. Now I'm pleased to invite Professor Wang to present your work as well as giving us an insight about the future research. Please, Dr. Wang, you may share your own. Yes.
Can everybody see my slides?
Yeah.
Yeah. First of all, I just want to thank Iskandar for the kind introduction. And I was very glad to forge this collaboration between Boston and Indonesia with Siri, Iskandar, and Harry. 3 labs, I think altogether we can do some amazing science together. So we met last year. So our collaboration was a little bit postponed by the COVID-19 pandemic. But again, this pandemic teaches us the importance of basic science, particularly the role of medicinal chemistry, medicinal plants. I'm sure the more we study, the more we'll be better prepared the next time we're hit by infectious disease like this the next time. So I look forward to visiting you physically, hopefully in the coming year. So today I'm going to present research that's ongoing in my lab. The title of my talk is Harnessing Plant Specialized Metabolism in the Post-Genomic Era. For students in the audience, so if you have questions, you can either put in the chat box and I'll catch them, or you can also ask them after my talk today. So research in my lab is very much inspired by this amazing natural diversity that's surrounding us. I'm sure in Indonesia, which I haven't visited, I think on a daily basis, we're basically living in this amazing diversity. I was very much intrigued as a kid, but as I grew up, I realized there's another layer of diversity known as chemodiversity. So in plants in particular, there are millions of molecules that are made by these natural chemists. And since plants are sessile organisms, that means once they're seeded, they can't move around like animals. So they have to deal with the stress around them through this chemistry. So chemistry is really the most important language that the plants speak to their environment. And I would say we only know the tip of the iceberg at this point. We also know plant chemistry is very important for human life. We almost enjoy it every day, ranging from drinking coffee and tea in the morning, using plant material as spices. We also use plants as energy source or as building materials or materials like rubber and wood. Humans have also used medicinal plants for thousands of years. In the last 100 years, some single molecules from medicinal plants have been isolated and identified. Many of them became single molecule medicines that we now use in the clinic. So on this slide, I summarize some prominent examples. For example, here, morphine coming from opium poppy is the first modern medicine that was promoted to be a very important painkiller in 1906 in the United States. Many compounds, as listed here, paclitaxel from Pacific yew tree, vinblastine from periwinkle plant, and many other are first-line chemotherapy drugs. And they have very elaborate structures, and since their approval are still being widely used today. So there are some small molecules, and the last two as illustrated here, huperzine A and icroside 2. So these could become very promising medicines, but they're coming from endangered plant species. And with some other reasons, for example, the patentability of natural products in the recent years, these compounds didn't make into the medicine list. And in the later today, I will talk about some of the bottlenecks in the process of making the plant natural products into medicine and how we can address them, these bottlenecks, through modern studies. I also want to talk about a little bit about sustainability, which is a big thing, not only in certain countries, but impacting the whole entire world. So this is a picture showing Pennsylvania in 1859. So George Bissell and Edwin Drake discovered the first oil rig in Pennsylvania. So this marked the beginning of the fossil fuel industry, and which is a very important part for the modern society since Industrial Revolution. And just within 150 years, now the whole entire world relying on the fossil fuel industry, the oil and the coal coming out of the ground, not only supporting our energy need, but also support the whole entire chemical industry, accounting for about 50% of world's GDP. Many of the industrial products, all the plastics, and a lot of consumer products, even including the pharmaceuticals we take, have raw materials coming from the oil refinery industry. So the whole entire human society has become reliant on the fossil fuel industry, which we all know it's not sustainable. The consequence of that is dire. So now we're living in a modern society, but now we're also facing a drastically changing climate, which is caused by rapidly rising CO2 levels and other greenhouse gases. So this is a very surprising rise of CO2 level just in the last few decades, which is entirely caused by human activities. So in many parts of the world, we can already experience climate change. So this is a picture showing in the Arctic regions. So these are permanently frozen lands, and there are very large sinkholes being forming because of climate change. And the process in turn release great amount of methane as CO2 back into the atmosphere and further catalyzing the climate change. So it's a rapidly developing situation which will impact every one of us in this coming century. So we all need new technologies to reverse this trend. So nature is a great resource to look for these inspirations. So now I'm going to show you a few examples with things that nature evolved versus the compounds or materials man-made. So on the left, we're looking at glue. And on the left, this polyvinyl acetate is a polymer that we use as a glue material. And in plants, they make much more complex polymers. So this is one example called the sporopollenin. It's a relatively enigmatic plant polymer that protects the vulnerable gametes of plants. And my lab was involved in solving its structure, which was published in 2019. So if you compare how humans make polymer and how plants make polymers, definitely we would say that plants are much more profound chemists. The similar trend also are observed in medicinal chemistry. So scientists have surveyed the chemical diversity within the drugs that have been proved since 1981. So if you look at the drugs that are coming from natural products, as shown in panel A, or natural products derived, their structures are way more diverse compared to chemistry that's entirely dependent on chemical synthesis, as shown in D. So here are some other examples. So I mentioned Taxol from Pacific yew tree, and it's a very complex molecule with many chiral centers. So that means this molecule is three-dimensional. In contrast of imatinib, which is Gleevec, a very important cancer drug. And as you can see, this Gleevec molecule is entirely flat. So compared to plant chemistry, we are still at infancy in terms of being able to create very complex molecules. So to this point, most of the compounds I've talked about coming from plants are known as so-called specialized metabolites. So they're conceptually different from the primary metabolites that many of you have learned through class. So on the left, I'm showing the TCA cycle, which is a fundamental pathway involved in generating energy. And most of the primary metabolic systems are conserved across kingdoms of life, and they're immediately required for survival. That means if you have mutations in any of the genes, you will lead to a dead organism. So on the right, I'm showing the specialized metabolic pathway for making Taxol. So these pathways are only distributed in certain lineages of plants, sometimes even in a single species. And these pathways can be very elaborate, and these enzymes are quite specialized. That means they're not found in other organisms. And if you have a mutation in these pathways, usually the plants will not die. But the consequence of that is a compromised fitness in their nature surroundings. So making these pathways quite different than the primary metabolism, eventually impacting how the ways they could have evolved over a long period of time. So over the years, the field have put a lot of effort in trying to understand the evolutionary process, how this remarkable chemodiversity could have come about through hundreds of million years of evolution in plants. So there are few conclusions we made from that investigation. So the first thing is most of the enzymes involved in plant specialized metabolism have their closely related homologs already existing in primary metabolism. So here is one example. So I'm showing two enzymes here. On the top is the enzyme involved in fatty acid metabolism called the Cas3. And at the bottom is the chalcone synthase, CHS. It's the first committed specialized metabolic enzyme in flavonoid biosynthetic pathway. They share very similar structures. Just with a few mutations, the primary metabolic enzyme can now evolve into a specialized metabolic enzyme and do something different. So this process have happened extensively through the last 400 million years of plant evolution. So in any of the plant genome that we have sequenced, there's a very big fraction of genes that are dedicated to secondary metabolism. So here in the middle, what you're looking are the phylogenetic trees for different enzyme families. Some enzyme families are very big. So this red one is cytochrome P450s. So humans also encode a few cytochrome P450s, mostly involved in detoxification of xenobiotics. But plants, each plant genome has over hundreds of P450s. Many of these are involved in making specialized compounds. So in diverging plant lineages, there is this independent radiation of these enzyme families, and the assembly of different offspring enzymes in a lineage-specific manner lead to the diversification of the chemical profiles now we see in different plants around the world. The other concept about plant metabolic evolution is the idea of enzyme promiscuity. So when we learned about enzyme catalysis in textbook, we often have this impression that enzymes are very perfected machineries. They do exactly the defined conversions as they're supposed to do. But if we examine enzymes in great detail, we found it's not the case. Just take this enzyme E1, for example. 99% of the time, it's converting A to B. But if you look very carefully, it also has this side reactivity to convert C to D. Maybe C is similar to A, so that this enzyme can also recognize it as the substrate. So when conditions change, for example, temperature or the pH in a cell, maybe this latent activity can get amplified. Oftentimes, if mutations happen to the gene encoding this enzyme, this side activity can be further amplified. If there's a gene duplication event followed by positive selection, then the original copy can keep the original function, which could be important, where the newly derived function can then become the major function of the newly derived enzyme, especially if D provides some sort of selective advantage to the host organism. So this is evolution of a new enzyme at a single gene level. And as you can see, the progenitor organism might have a relatively elaborate existing pathway, and this process could be happening all the time. So then you have multiple newly evolved enzymes that are derived eventually may form a new pathway, making new chemistry. So this is kind of how we view metabolic evolution in plants at both the genomic and pathway level, as well as at the single enzyme catalysis level. So these days in the lab, we use a multi-omics approach to study plant secondary metabolism in a wide range of so-called non-model plants. So this was not possible 10, 15 years ago without the advent of next-gen sequencing technology and some other types of omics technology. So we could now study any plants of interest. Here is a very typical workflow for that study. So when we get that plant, we would sample it very finely. So for a typical plant, we will sample the leaf, the flower, the roots, the stem, sometimes different tissue types of the roots or of the flower. And then we divide the samples into two parts. The first part, we do RNA-seq, so just to capture all the genes that are expressed in the tissue. For the other part, then we'll do mass spec-based metabolomics, so that hopefully we can capture a large fraction of the metabolites that are present in this tissue. So then we can do correlation analysis and trying to find the genes encoding the enzymes that may be responsible for making the molecules of interest in that tissue. So here is one example. So this is marijuana plant. It's legalized in many states in the United States. My lab doesn't study this, but it makes a very good example for this approach. So the cannabinoids, this is the class of molecules that are responsible for the medicinal properties of the plant, are very much enriched in the glandular trichomes. So these very nice looking cells on the surface of the leaf. So if you were sampling these cells and then you sequence what are the genes that are expressing these cells, you find they're very much enriched with cannabinoid biosynthetic genes. So this has proven to be a very effective way to elucidate unknown metabolic pathways underlying the synthesis of many medicinal natural products from plants. So once we have gathered a list of candidate genes, then we actually take approach known as synthetic biology to trying to put the pieces together. So for many of you, when you think about synthetic biology, you might think about this as an engineering tool. It's something that once you understand a system, you can now engineer. but I would argue that you can actually use this as a discovery tool. So I'll quote Richard Feynman here. He said, "I cannot understand what I cannot build." Let's say I have in my hand 20 candidate genes, maybe only 3 of them are required for making this hypothetical molecule of interest. So if I'm trying to build them together, once I found the right 3 genes, just putting them together in a heterologous system, the moment I can make this molecule is the moment I have realized I've discovered this unknown pathway. So this is exactly how my lab is utilizing the approach of synthetic biology to understand or to elucidate the pathway for unknown natural products. The other benefit of that is once you have done that, if you were, let's say, doing this in yeast, you actually have created this prototype of engineered microbes that can be further optimized to make high value products. So this leads to a whole new industry known as metabolic engineering. So through fermentation, which is well-established industrial process, you now can make high-value plant natural products by adding additional genes, which not necessarily from the same plant host. You can further modify these molecules to create so-called new-to-nature molecules, which could be patented and made into either medicines or commodity chemicals. So that's a very exciting new workflow to how to study medicinal plants. You start from a plant of interest, you figure out their genetic makeup by doing next-gen sequencing, and you can also do a metabolic profiling by linking genetic information with their metabolic profiles. Then you can find the genes responsible for the synthesis of compounds of interest, and you try to engineer this process in other organisms that lead to newly engineered organisms that can reproduce these molecules, and even newly derived molecules. And these could be very useful for a variety of downstream industries. So over the past few years in my lab, we have been working on several medicinal plants. Some are illustrated here. So these are quite exciting exercises. I'm not going to go through each one of these today. So I will just spend time on 2 examples: the Kavakava plant, which produces anxiolytic compound called Kavakava, and in Goji berry, which produces cyclic peptide called Luciomin. So besides studying medicinal plants, I also mentioned a little bit about plant polymers. My lab is also quite interested in how plants manufacture structurally elaborate polymers. So spiropollenin is one example here. We are also, especially in the recent years, looking into ways to engineer other polymers such as cellulose, lignin, and suberin, and making them decay resistant. So we believe this could present a new methodology to carbon capture as a way to reverse climate change. So I will talk about this first example of Kavakava, and this is a very interesting plant native to Polynesian islands. The plants only grow on these islands and have very specific requirements for the growth condition. So I actually tried to grow this in Boston climate, and it really failed miserably. So it like tropical conditions. So the plant has been consumed as a tea, as shown here, by the native people living on the island. So it gives people a very calming effect. So I think it was discovered at a very early times when these native people migrated to the islands. And drinking this plant has become a central element of their native culture. So this is when Pope and Prince coming to visit Fiji, and the local people will treat them with the kava drink, which is a very high honor. So I learned about this story probably 3-4 years ago when I went to listen to a seminar at Harvard Museum of Natural Sciences. And I found it very interesting. Here is also to mention the Kava drink is becoming very popular in the United States as a safe painkiller among athletes. So when I heard the story, I found this quite amusing. So I looked on my phone. So I found the chemistry for the Kava plant has been solved at the time. People know Carbohylactones are the main bioactive compounds in the plants, which count for about 20% of the dry weight of the root, which is quite remarkable because you think about plant material, it's usually the fiber or the lignin or cellulose. For a set of small molecules to count for that much weight, that means remarkable selection has occurred in the history of these plants, promoting these plants to make a lot of these compounds. So because of that, a person could intake more than 2 grams of Kavakava if they go for 1 drinking session a day. And these people are drinking these on a daily basis, suggesting it must be safe, a very important feature if you want to consider as medicine. So the plants also make some other natural products, for example, flavonoids, which are flavonoids. These molecules are interesting on their own rights. For example, they have anti-inflammation activity or anti-cancer activities. Some are also known to target molecular chaperones like HSP90. So when we look at the carbohylactone structure, compare these to the flavonoid structures, the biosynthetic hypothesis starts to emerge. So at that point, we knew how typical flavonoids are biosynthesized. So I showed the structure of CHS, which is a conserved enzyme, exists in all land plants. So flavonoids are made through the CHS enzymes through this polyketide synthesis mechanism. So for making plant type 3 polyketides, you start from a carboxylic acid, in this case, cinnamic acid or p-coumaric acid. You activate that by linking that to a CoA thioester. And the polyketide synthase, CHS, in this case, it's a tetraketide synthase. So it produces a tetraketide intermediate. And this tetraketide is high energy. So within the active site of the enzyme, it'll cyclize, and then through Claisen condensation will make into the basic backbone for flavonoid. So from this point on, there are a lot of structurally diverse flavonoids can be made. But when we look at cavalactone, it's a lactone. So then we hypothesize instead of going through a tetraketide intermediate, kava plants may have a styrylpyrone synthase and goes through a triketide intermediate. And the triketide can then cyclize to make a lactone ring. So this is the kind of the hypothesis we were forming when we examine the structures of cavalactones, comparing that to flavonoids. So then we obtained the kava plants from Hawaii. For the period of time they survived in Boston, we did enough experiments. So we sequenced the transcriptome of kava from the root tissue, and we identified not only CHS, which has to be there. Very intriguingly, we found only in kava they have 2 additional genes that look very similar to CHS. So in this case, we would call them the paralogs of CHS, and we name them SPS1 and SPS2. Both of these genes are highly expressed in the roots, shown here with very high TPM value. These are the quantification matrix from the RNA-seq experiment. And this particular branch also shows signs of positive selection. And this happens only in the kava plant, because if you look at it's closely related plants in the same genus, the Piper genus, and piper beetle and pepper nigrum, so this is black pepper, they do not have these events. So this gene duplication event followed by new functionalization that gave rise to these two genes happened after the Kava plant diverged from its last common ancestor. So this is a trait that would for sure entertain Charles Darwin if he lived to look into this. So we were able to clone these genes, and then we expressed these genes in E. coli, purified the recombinant proteins. Then we assayed the biochemical activities of these two genes. We found that indeed SPS1 and SPS2 are not CHS enzymes. They have evolved novel functionalities to make the lactone, bisnoryanganine in this case, as their main product. So we then went on, solved the crystal structure of SPS1 and comparing this crystal structure with the CHS crystal structure. What we realized is that there are three very unconventional mutations that have occurred that very subtly changed active site shape and volume of SPS1, leading to this novel reactivity. So as we know, it's a polyketide synthase. If you restrict the volume of the active site, instead of generating a tetraketide intermediate, the smaller active site only allowed the triketide intermediate to be generated. So using very similar approach, we were able to identify some additional enzymes, in this case, a very key reductase that generate this signature sterol center in the carbohylactone backbone, and also a methyltransferase. If you if you methylate, then the reductase can still proceed. But if the reductase works on the precursor, then the methyltransferase will not proceed. So the combination of these enzymes generate further diversity around this carbohylactone backbone. By the way, we also solved the structure for the reductase, resolving its structural basis for generating the sterol carbon center. There is also a P450 enzyme that's involved in producing this methylene dioxide bridge, which is found in other natural products. So with all these enzymes, we now can we resolve the carbohylactone biosynthetic pathway in Kava, and now can produce up to 10 naturally occurring carbohylactones through microbial fermentation. So we also put into the systems halogenases from bacteria that allow us to further derivatize carbohylactones. Now we're making a set of new to nature carbohylactone analogs, which become very promising in terms of downstream pharmaceutical development. So yeah, so this is Tomasz Blaskel, the postdoc who was working on the project. And this is the first week when he bought Kava powder from Amazon and drinking with his wife. And the second day he reported it does work. And surely we have tested Kava ourselves. We had drinking sessions at the Whitehead Institute. It does have very profound calming effect. So I want to mention this point is many of the medicinal plants have been practiced in human society for thousands of years. We know their properties, medicinal properties, and if we use them properly, the safety can also be guaranteed. And we just need to figure out the mechanism why they work. So I call this reverse phase clinical trial. So we know it works in human, and then we move on and do tests in animals and trying to figure out the medicinal mechanisms. So in this case, we use zebrafish as our experiment model. So if we know that if you add Kava extract onto zebrafish, it will stone the fish. So it's kind of similar as people drink Kava. You will feel a little bit sleepy and dizzy, and you just want to sit down. And here, as you see this fish and has difficulty maintaining its balance, but if you retrieve it from water and put it in regular water, it's actually okay. So this is our working model. And at the Whitehead Institute, I collaborate with Hazel Siff's lab, who is an expert in zebrafish, and she uses fish to study neurological disorders. So you can really observe fish behavior under a video camera and do quantitation on this. So prior research have indicated fish being a very good model for studying anxiety. So when fish is anxious, it tends to swim around a well. So this is a well of a 24 well plate. And then if the fish is anxious, it tends to swim around. If fish is calm, then it tends to stay in the middle. So if you add molecules to the well, and then you can then monitor the fish, then you can quantitate how calm the fish can be. So that's exactly what we did. We add defined concentration of different Kavakava to the well and monitor how the fish would react to this treatment. So the answer is quite remarkable. We found all 6 Kavakava change the fish swim behavior called sigmotaxis. All 6 Kavakava can reduce the anxiety levels in the fish, whereas the control flavonoids and caffeine cannot. We found yangonin, which is this molecule here, has particularly good activity towards reducing fish anxiety level. And now we're moving towards developing this further as a candidate drug for treating human anxiety. So why these molecules work, and there are other studies from other labs suggesting not only Kavakava target the GABA receptors, which are their main targets in the CNS. Certain Kavakava, for example, yangonin, also target the cannabinoid receptor, for example, CB1. So making these molecules very interesting molecules that can regulate human mood and sensation from multiple receptors. So through this research, hopefully we can come up with new molecules that could help with many of the kind of opioid epidemic and also neurological diseases that affect many people around the world. So the second story I'm going to jump into is on a set of molecules known as ribosomally synthesized and post-translationally modified peptides, or RIPs. So these are quite interesting molecules that are mostly found in bacteria and fungi, and there are a few of them have been reported from plants. And through our study, we found the RIPs diversity is very much underexplored in plants, and which contains not only very interesting medicinal chemistry, but also unexplored biochemistry that supports their synthesis. So the particular sets of molecules I want to focus today are known as Multiomics. So these are known as branched cyclic peptides, and they were initially found in Goji berry, which is a medicinal plant in China. So these compounds have 2 unique structure features. One is the N-terminal pyroglutamate structure, like here. The other one is this conjugation that forms the heterocycle. So it's different from the typical NC peptides. The cycle forms between the tryptophan indole nitrogen with a backbone carbon. So that these 2 features give the characteristics of the branched cyclic peptides. So when they first were isolated, they were shown to have very good inhibitory activities against human angiotensin converting enzyme. And many years later, we now know ACE is actually the receptor for the SARS-CoV-2 virus. So knowing this, we know maybe some compounds from goji berry could be antiviral. So RIPs are, as the name indicates, are produced from the ribosomal protein. So this is a very typical biosynthetic mechanism for RIP synthesis. So its precursor peptides are encoded by the genome. And when the gene is transcribed, translated into the precursor protein, there are multiple steps of modifications that have to occur. So usually there is a typical post-translational modification that forms the cycle, that defines the structure class. Then there are proteases that would follow, that cleave off the leading peptide or the C-terminal downstream residues to produce the natural peptide. So bearing this in mind, we did the transcriptome mining of goji berry. We sequenced the transcriptome of goji berry roots. From that, we identified a gene of unknown function that's ranked very high in the transcriptome. So it's the number 13th of the most highly expressed gene in the goji berry root. So this gene encodes a protein that has the N-terminal end have repetitive motifs that have the sequence corresponding to different leuciomine peptides, as I showed in the previous slide. And this gene also encodes a C-terminal burp domain. At the time, really nobody knows what the function might be. So when we know this, we kind of predict this is the precursor peptide for leuciamine synthesis. Then postdoc in my lab, Roland Kirsten, did this quite remarkable experiment. So he took this one gene and transiently expressed this gene in tobacco plant, Nicotiana benthamiana. And just by expressing one gene, he was able to produce leuciamines in tobacco plant after a couple of days. And then that indicates this is the only missing piece for leuciamine production, whereas all the other machineries, whatever they are present in the tobacco plant. So with this initial discovery, then Roland went on to do and did a gene-guided chemistry discovery in wide selection of plants. So he took the leuciamine precursor peptide as the query and did blast search against all the sequenced genomes. And in that exercise, he found 21 out of the 116 sequenced genomes contain genotypes for leuciamine. And he then went on to verify the presence of the predicted leuciamine chemistry by metabolic profiling. So he found there are 3 families of plants, Amaranthine ACA, Fabiaceae, and Solanaceae being the 3 most productive families for leuciamine chemistry. Very interestingly, many of these are crop plants or vegetable plants. In closely related plants, for example, these 2 different type of potatoes, they actually generate different leuciamine chemotypes. The russet potato, the cheap potato, generate the leuciamine J, and the red potato, the more expensive potatoes, generating leuciamine B and K. So giving us a lot of peptide chemistry to explore for their bioactivities. So this exercise, we also learned the structure-function relationships for leuciamines. We know in the structure, there are certain residues that can vary among different residues. Certain residues are more conserved. In terms of the position of the core peptide, in Fabiaceae, the core peptide motif is embedded within the burp domain, whereas in Amaranthine and the Solanaceae, the core peptide is outside of the burp domain. So, and through the study, we also laid out the biosynthetic pathway for making leuciamines. We know that the post-translational modification to form the ring happens first. Then the endopeptidase follows to cleave the N-terminal end. We identified a glutamine cyclotransferase that convert this N-terminal glutamine into pyroglutamate. And then we have the exopeptidase, then does the C-terminal cleavage to produce the final peptide. So there are many pieces of evidences supporting this proposed pathway. And I'm not going to detail it here today. And many of the genes are also clustered in certain genomes. For example, this precursor peptide is just next to the glutamine cyclotransferase in the quinoa genome, as shown here. We have some evidence showing the first cyclization reaction might be catalyzed by the burp domain itself, but we're still in the process of looking into that. The one benefit about peptide natural products, especially RIPs, is that the diversity of chemistry is encoded directly by the genes. So we took advantage of that property, and we identified one particular precursor peptide from soybean. There's only one core peptide motif without repetition, so making this very easy to be manipulated. And we were able to do alanine scanning mutagenesis and show that there are 5 residues out of the 8 residues that could be modified. So that means, in theory, we can generate over 3 million of different peptide chemistry using this one gene. So this really will going to be a very powerful approach for synthetic biology, and especially for screening for bioactive peptides for medicinal uses. We also found that if you vary the repeat numbers of the multipeptide, you can change the ability to synthesize, or you can scale the production of the final peptides. So in this experiment, if we introduce 1 copy, 5 copy, or 10 copy of the motif peptide, then you can really scale up the production of a particular luceamine peptide in transgenic tobacco. So this is, if you look at this very tall bar, you can compare this to the 4th bar, which is the native production of the peptide from goji berry. So by engineering the whole process in a heterologous plant, we can really scale up the production of these medicinally important peptides. So just to summarize of my talk today, why we're here doing this at this time, because there has been some very exciting development in multiple areas of research in the genomics. Our knowledge about the evolutionary principles of plant specialized metabolism, the advent of synthetic biology and metabolic engineering methodologies, and also from the chemistry field, there are also rapid growth of transformative analytic chemistry techniques, such as metabolomics. So combining all these techniques now that allow us to really explore the historically underexplored medicinal plants and utilize them to create, recapitulate natural products and so-called new to nature products. So these would really benefit future industries for pharmaceuticals, nutraceuticals, and other commodity chemicals. And I want to acknowledge my lab. Many people contribute to the studies I just presented. I would really acknowledge Tomasz Plaskel, who was a former postdoc in my lab, who did the Kava story. He has recently started his own laboratory in Czech Republic. Former postdoc Roland Kirsten, who did the Rusumin story, and he also started his own laboratory at University of Michigan, Ann Arbor last year. And many other people contributed to the story, and also here are the funding agencies. Thank you very much again for the invitation. I'm very happy to take any question that you may have. Thank you so much.
Thank you, Jinke. Just 45 minutes. I noted very good time management as well as very great presentation, presenting about the relation between biodiversity and chemodiversity, and also the new methodology or approach, yeah, using the multi-omic technology. And you also presented very interesting case studies on at least 2 or 3 plant species producing taxol, and the new one is Kava, I think, and then the last one is about human. And now we are entering the discussion session. I'm checking from the meanwhile, can you stop sharing, Jinke? There is one question. I would like to invite here the first question from Dr. Sri Rahayu. So Dr. Sri Rahayu is a research scientist at the Bogor Botanical Garden. As you may know that this botanical garden is very old, yeah. I think 2 years ago they have an 200 years anniversary. Yeah. I would like to invite Dr. Sri. Please, can you ask directly as the first question to Professor Jing Keweng and introduce yourself, of course.
Okay. Can you hear me, Pak?
Yep.
Yes. Okay, good.
Okay. Yeah, thank
so
you.
Your background is botanical garden, yeah?
Yeah.
It's very beautiful.
Very beautiful.
Yeah.
So we missed the visit,
Okay.
Professor Jinke.
Okay, thank you. I'm very happy to hear your presentation, Professor Ching. And thank you very much to Pak Iskandar who invited me to attend this event. And I would like to ask how I can use the effective sentence. I don't know, because I have case. I have my plants in Hoya group.
Yeah.
Hoya, yeah. And it has medicinal properties, and we already extracted the chemical compound of Hoya, and there are some sorts of chemical compound. We call it phytochemical. And after that, I have collaboration with IPB University and biology department. And we have master students who conduct research on endophyte. And we isolated some of endophyte, and some of them produce the same chemical compound as extracted before from the plants. And so I conclude that the extracted chemical compound from the plants is really the produce from the endophyte. It's bacterial endophyte. And after I hear your presentation, so I have a question: is it possible if there is in quotes, a collaborative metabolism between plants and endophytes, especially in medicinal plants?
Thank you.
Yeah, that's a great question.
Thank you very much.
That's
Yes,
exactly something my lab is very interested in, especially in the recent years. We have a very similar case as what you just described for another peptide natural product. So in our case, it's called the bovadian. So bovadian is an anti-cancer compound found in the Bavaria plant. So for many years, people thought it just the plant produces the molecule. We were a little bit suspicious because just by looking at the chemistry of that peptide, it looks different from something that plant would produce. There are some chemical features that are very similar to microbial peptides. So indeed, we identified it's one endophyte or maybe more that produces it. So there are a lot of unknowns. So I think these systems are extremely interesting. So in our case, we found this endophytic associations very specific because you don't see this microbe living in other plants. They have formed this very special relationship with its plant host, implicating that there are some signaling going on. So there's this signaling aspect that would be very interesting. So maybe the plants are supplying some either precursors or nutrients or some signaling molecules that allowing the endophyte to survive within the plant. And then the endophyte is producing these natural products. Their biological functions are usually not known. So I would suspect that these may provide a layer of defense for its host plant. So when these, as you said, the collaboration between a microbe and a host plant forged, they produce this ecosystem, which is largely unknown, that make both to survive better in their surrounding environment.
Okay, thank you very much.
Thank you, Professor Jinke. I have another question. So this is from our college from Sriwijaya University. Pak Arin Aflil, are you there? Pak Arin? Oh, I hope he is still there. But the question is, I just read the question. The current losses of biodiversity will affect ecosystem function and the ability of nature to provide goods and service for human health and well-being. To get benefits of natural products, biodiversity conservation should be taken into consideration. Talking about drug discovery from biodiversity, we have to consider as well the life of indigenous people, maybe which maintain the collectively the local knowledge and give respect to their knowledge in biodiversity benefits. So his question are how to raise awareness to gain economic benefit by protecting biodiversity for ecological aspect with risk of habitat destruction in one side and wisdom of indigenous people in another side. So this is about the maybe regarding the access and benefit sharing between scientists or industry as well as the local or indigenous people. Can you respond it?
Yep, it's a great question. It's a very big question. I'll probably just share some of my thoughts. I think in the early days, people didn't do it ethically, right? So there are, the age of exploration when Western culture, after the Industrial Revolution, became stronger than the rest of the world, then they explored the world and they took whatever they saw. And, sometimes they spread disease and they kill off native people. So a lot of horrible things have happened, which occurred until maybe till the 70s and 80s before there is enough awareness that the biodiversity has to be protected. So I think at this point, I'm sure in Indonesia that's the case, but I know better, for example, in China or in Japan, you can't just freely enter a country and grab whatever species you want. So most of the countries have established laws to protect their native biodiversity. But through legal channels, if you report this is something you want to research, I think these collaborations can be formed. So it's paid with tremendous respect and the sense of protection. So I think on that regard, it's much better than before. So that's first. Second is, I think, basic research and also translational research from that is a form of conservation, right? If we do not really understand why these plants have medicinal values, it just remains as a traditional wisdom that may benefit local people, but that value cannot be fully released. So by researching the plants first, we can preserve its genetic makeup. Let's say, samples of this are preserved, or seeds of these could be preserved in research institutes. And if we sequence the genome, even maybe after 100, 200 years, if the climate really went to pretty bad situation, then the plants are wiped out. We have a data copy of its genome. Maybe we can resurrect these plants that went extinct. So as the process, as I just described, once you learn the pathway, maybe you can then produce the same natural products without having to sacrifice the plant. So let's say some of the very valuable medicinal natural products are from those trees that have to grow for 200, 300 years. It's just not feasible to cut them down and then use them for extracting medicine. But if we learn how the plants do this, we then engineer a yeast strain that produces the same molecule, or even we identify, just as the last question, there's an endophyte, so we can isolate this endophyte, we can do fermentation, just use that endophyte, we can produce the same molecule. So by doing very much in-depth study, we have achieved a very effective conservation method. So that's the second point I want to make. And the third point is this kind of overwhelming threat coming from climate change.
Yeah.
When I was a little bit younger, I thought, and also in the media, it says it may play out in 100 years, maybe 70 years. It's essentially out of the scope of our generation. You know, for people sitting in this audience, by 100 years later, most of us would not be here. So a lot of us would think this is so far away. It's something for the next generation to worry about. But just in the last 5 years, I think we're seeing a kind of acceleration towards disaster. So I don't know about you guys. This year in Boston, in the past few years, we have been having very warm winters. And in this year in the United States, there are a lot more wildfires in states like California, Colorado, which are very unusual. And we had a very historically dry season in Boston in the summer. So these are quite unusual. I think the fire itself, it burns, I don't know how many gigatons of forest being destroyed, that carbon becomes released to the air. So the impact is not local. I think when the globe, the earth is warming, you know, I'm sure Indonesia will be affected. Other places like Africa, maybe it's on the verge of, you know, barely sustaining its population, but with 1 or 2 degrees of temperature rise, that piece of land would not be able to grow any crops. So this is it's quite a pressing issue that facing humanity, and collectively as a whole human beings, we really need to develop all kinds of technologies, first to remove us or to eventually shift us to renewable energy sources. And second, we need more technologies to reduce carbon emission. So that's a very important aspect of preserving biodiversity, which we really treasure, which are so beautiful around us. We don't want that to be destroyed because we are so irresponsible for the environment.
Thank you, Professor Jinke Weng, for the 3 excellent comments. So we learn as well that, yeah, very important to note that our basic research is part of the conservation efforts. Otherwise, we don't know what to be conserved. That's not wise, I think. Yeah, I still open to the last question. I think I would ask here from the audience. I note there is many researchers, participants. I would like, for example, here invite Professor Irmanida Batubara or other volunteer to give a question to Professor Weng. Bu Prof. Irmanida, are you there? Or I saw here Dr. Deden Drajat. Do you have a question, Pak Dr. Deden? Because you are doing also transcript. Oh, Bu Irmanida first, maybe. Any question, Bu Professor Irmanida? I met you. She met you also at your lab, Dr. Irmanida. Please, Bu.
Yeah, thank you, Pak Iskandar. Hello, Prof. Jinke. We met again, but by virtual meeting only. So the question for you is, since as you know, Indonesia has a lot of jamu.
Jamu, ya.
Yeah, a lot of jamu or Indonesian traditional medicine.
Yeah.
Can you give us suggestion how to prove that our traditional medicine is effective?
Yeah, that's a great question. Very practical. So I can speak to how this is done in the US, for example.
Yeah.
So in the US, I think it has the earliest government agency established to regulate food and medicine. So this is the FDA, the US FDA, the US Food and Drug Administration. And I mentioned a little bit in 1906, morphine from opium poppy became the first approved modern medicine. So that's a single molecule medicine. But FDA, since it has established, actually remained to be extremely simple in terms of establishing something that will be therapeutic. First, it has to be safe. Second, it has to be effective. Whatever you put forward in front of the agency, as long as you can fulfill these two requirements, then this is considered scientifically as a bona fide medicine. So many vendors, developers come with small molecules, proteins, antibodies, vaccines, but there are also small fraction of the people come up with plant mixtures. So there are I think two plant extracts that have been approved. Yeah, so one is dragon tree extract, which is for treating a topical skin condition. I think the other one is green tea extract, also for skin condition. So as long as you can show that every single dose you're providing to your patient has pretty much stable chemical composition, you can guarantee the quality, you can show safety, and in double-blind controlled clinical trials through 3 phases, phase 1 to 3, you can demonstrate efficacy, and the FDA can then prove this is a really useful drug. I think it's a very high bar to other countries because I was born and raised in China, and China is also very rich in the use of herbal remedies. It has its own system, and I do know it works. It's not, I won't say that if any compounds or any herbal remedies not going through clinical trials are not useful. I would never say that. But I think if you want to convince other cultures or maybe out of your local, use communities that something you are working on is a drug, is a useful medicine. And these are 2 criteria that as a community or as a country we should consider to be safe and efficacious through human clinical trials.
Okay, to be safe and efficacy,
yeah,
very important. Okay, so still maybe one short question, Pak Deden, are you going to ask something? I heard that you are research on mangosteen, yeah? You want to say something?
Yeah, so I just have one question because I'm interested with the gene regulation in form of the specific compound in the plants, because I am now to study some gene regulation to affect the isoform of the gene that you mean in your last slides, that the repeat region, the repeat sequences can affect the concentration of the compound. So how to elucidate the compound is formed, that if many isoform in the gene, and also the isoform that affected of the concentration of the compound.
Okay,
yeah,
that's the last question, I think. Please.
Okay, hopefully I catch your question. So in this particular case, it's quite unusual because, as I said, the Ribosomal peptides, the chemistry is encoded in the gene. So then if you have 1 copy in 1 protein, or if you have 10 copies in the same protein, then you can definitely greatly increase the amount of final products to be produced. But that's an exception in natural products. I think for many other natural products, for example, Kavakava, they have to be synthesized by enzyme conversions. So in this case, the final product concentration is dictated by several different factors. One is the availability of the precursor. So let's say if the precursor was one of the 20 amino acids, so you need that to be abundantly present for the final products to be made. Second, a lot of time, the production of certain natural products is induced by environments. So pathogen attack, insect chewing, or even condition, cold temperature, or some drought stress can also elicit the production of the natural products. So these are the more common situations you would need to address. So if you are looking at a particular plant, a particular natural product, you probably would like to figure out what would be the best condition to elicit the production of this natural product, and in which tissue this product is being made. So there are many questions that you can ask, but if you are interested in elucidating the biosynthetic pathway, this is quite important, because if you can elicit the production, then you're better off in terms of finding the candidate genes for that process.
Okay, thank you.
Good, thank you. Now we are moving to the next sessions. Key takeaways or key message to be brought for us in Indonesia. Since you might know that the Indonesia is relating the question to Dr. Arin Afril from Sriwijaya University, that Indonesia ratified the Nagoya Protocol in 2013, yeah, as a law. So this is this protocol is deal with the access and benefit sharing if we are accessing the genetic resources and traditional knowledge related genetic resources, as been discussed, and you also already elaborate, Professor Jin K. Weng nicely. So I would like to invite Ibu Sri Fatmawati. I observe that you are very busy noting key important issue from the presentation as well as discussion. I think you have maximum 10 minutes to, let's say, kind of wrap up or for us as a researchers, the takeaways message. Please, Ibu Sri Fatmawati. So by the way, before you are going, so I just would like to congratulate Dr. Sri Fatmawati as the President-elect of Indonesian Young Academic Science. Yeah, yeah, you may. Yes, Bu, please.
Okay. Okay, thank you, Prof. For your nice introduction. I would like to thanks for Prof. Jinke for your very good presentation for us. So I think all of the participants here have the very clear insight about how what we can do for our biodiversity in Indonesia. And as you mentioned, that the vast chemical space existing in nature is largely unexplored. So I would like to share with you, all of you here, that one of our, since I belong to the Indonesian Young Academy of Science, which is also belong to the Indonesian Academy of Science, we have one book, namely Science 45 or Science 44, how we, how we become, what we do for the 100 years of Indonesian independence. So in that book comes from 100 young scientists throughout in Indonesia. We try to make give our talk, our ideas, how to make Indonesian better. And one of the chapter in the book is about life, health, and nutrition. And we start, and where we start. So we finally choose one of the ideas is biodiversity. So we start from this biodiversity because we know Indonesia is one of the biggest biodiversity in the world. So this topic today, you talk about the how harnessing plant specialized metabolism in the post-genomic era is very related with our, our resources. And one of the, I think one of the tasks here that we have to notice here is demystifying of natural products. So we still have a lot of folk medicine, and we still have a lot of local wisdom that we know. We still, we still have maybe zero knowledge of it. So your talk is a very important to be part of our innovation, to be part of our inspiration, how we can clarify our Indonesian natural product, our Indonesian traditional medicines, and also others. Yeah, and here, to be honest, I enjoy very much your talk, and I wrote all of the things, but you mentioned about the how we can sustain and how we can still protect our endangered species, and because in Indonesia we also have the same things. Yeah, and I notice here that one of our Kita's message for all of you is the biosynthetic capacity of nature is far away exceeds the world of the best synthetic chemists. So, and also when I learn about your research, your talk about the Flavonoids, yeah, and also what is it? Luciomin, right? So I think that Indonesia also have the same, but still unexplored. So, and as the your presentation said that the combinatorial biosynthesis of how we create nature and natural-like products is very important message for us. Yeah, and we can, I do believe that we can extend chemical diversity beyond the nature that already offers by using science and technology. And that finally we can use it for the pharmaceutical, nutraceutical, and also other commodity chemical. And I noticed here also that we as a natural product chemists and also as biologists and also scientists, that together here we can do something for Indonesia, and also not only for Indonesian people but also for the world, because if we can collaborate between science and natures, the unexplored nature, we can do better. So I think that's all for me. So we try to, we have a dream in the book of book 45, Science 45, the book of Science 45. One of our dream there is how we can utilize, how we can, harnessing the Indonesian natural product for the humanity. So I think that's all for me. Prof. Iskandar, hopefully I'm not exit from the 10 minutes. So
okay, thank you, Bu. Very nice.
Yeah, very much for your talk today, Prof. Jinke. Yeah. And I know right now in Boston is
very late. Yeah. Okay, thank you, Bu Sri. Yeah.
Thank you.
Thank you, Dr. Sri, for a nice elaboration and also becoming also homework for us, especially the book produced by Indonesian Young Academy of Science. We will, I think it can be downloaded in the portal. So before we are going to the group photo, I would like to invite here Professor Heri Hermansyah. Pak Heri, are you listening to us? It seems the mic is a bit Halo Pak Heri. Prof. Heri Hermansyah. So maybe he has a problem in the connection. Prof. Heri Hermansyah, could you copy us? So until he just very shortly until he's showing up, then we might have a group picture first. So please, delegates and participants to turn on your video. I think Mr. Miftah will ask to turn on. Please to get the. I think we have here three. Miftah, can you start to take a picture? So we have 3 pages here, but it's okay. Please turn on. Oh, Pak Heri. Okay. Before that, please, Pak Heri, can you give a short closing remarks? Professor Heri is the director of intellectual property management of Kemenristek BRIN. Yeah. So you have 5 minutes, Prof. Heri, to let's say. Tell about the future works on collaboration and research, as well as specifically with the MIT or MIRA program. Please, Pak Heri.
Okay. Hi, Professor Weng. Good morning in Indonesia and good evening in US, I think. Okay, and Prof. Iskandar, all professor, lecturer who attend this Zoom meeting. Okay, at this time, for your information, we are still doing in major agreement, finishing major agreement between MIT and IPB, but the research funding has been already secured and funded by LPDP. And for your information, the ministry will support not only this program, we will support a similar program with MIT and ITB. For example, in the field of health and medicine, we also now in the process engaging engage collaboration between university alliance in Indonesia with some university abroad also. And this typical university in Indonesia and top university abroad will be driven by ministry to proceed in the same way. I hope this program will produce not only the collaboration itself, but can give outcome and impact, not only research output such as publication, but can give more outcome. For example, by starting communication and collaboration with Professor Weng, Professor Iskandar can deliver another program. For example, this program, workshop, and then after this maybe mobility between student, staff, and then after that research publication, and the outcome of the research publication, of course, the research citation, for example. And if the collaboration between the group of university in Indonesia with Professor Weng can deliver the intellectual property right, for example, the intellectual property right can give outcome, the IPR, the productive IPR, which can deliver the license or commercialization from IPR to be some product and economic based innovation, for example. So we must thinking about the research and innovation process in the whole, not only the research and the output itself, but from input, process, output, outcome, and impact. So I hope this collaboration will sure will continue. Yeah, will continue in more bigger group and can give more impact and more outcome for both sides. Thank you very much.
Thank you, Professor Heri Hermansyah. So a very good message, last message from him, and we hope that this community, at least we have their own address, then we can connect each other through the hub of this event. So now I give time to Pak Mas Miftah to group photo, yeah, session. Please, Miftah. Okay, how many slides? 3, yeah, 3 pages, yeah.
3 pages.
Okay, please tell us. Okay.
For first page.
Smile.
And the second one.
Second page. And then.
And the last page. Yeah.
Okay, thank you, Miftah. So I would say goodbye. Thank you for your participation. Thank you, Professor Weng, for coming to our event.
Thank you so much.
Thank
Have
you
a good
so much.
day.
Keep safe, yes, and then stay healthy, yeah. Bye-bye. Thank you, Bu. Thank you, Pak Heri. Thank you, everybody. Bye-bye.
Bye-bye. Thank you very much. See you again next time.
See you again next time. Bye-bye. Thank you. Bu Irma, oke. Thank you, Bu Irma. Bu Sri, selamat bertugas. Mbak Evra, Tanta dari Bengkulu. Pak Arin Afril ada di sini? Kayaknya tadi koneksinya susah ya. Oke, terima kasih ya teman-teman semua. Pak Jukruf.
Ada Pak.
Oh, Pak Jukruf, amin. Sehat-sehat selalu ya di sana ya.
Prof, terima kasih.
Siap,
Alhamdulillah.
siap. Ya. Oke. Pak Arif, Arin, oke, suaranya agak ini ya, gambarnya agak koneksi ya, masalah ya. Terima kasih atas pertanyaannya. Bye-bye.
Ya, terima kasih Pak Iskandar. Auf Wiedersehen.
Ya, auf Wiedersehen. See you again. Bye-bye.
Cheers. Bye.